Uh, is it okay now? Hello.
It's fine. Yeah, it's fine. It's fine.
Mm-hmm. After in a moment or so, I will be in Wi-Fi connection. So I hope that will solve the issue. Okay. So when you said about Prayagraj, what came to my mind is recently Mahakumbh happened, and people were talking about fecal coliforms, the load of fecal coliform after Mahakumbh. The load was quite high. It was claimed. Yes,
No,
Madam?
actually, but there was no incidence of E. coli, but high prevalence of Klebsiella, and there was Pseudomonas then.
Yes, exactly. Because they are quite common in our gut, so fecal coliform is one of the indicators of the water health or the pollution level in the water, right?
Yeah, exactly. Like the main source, what I studied from the research paper, that main source of contamination is itself the chickpea or whatever that sprout is made from, any gram or any lentils. And the second most prevalent contamination source is the water which they sprinkle every now and then. And then there is the obviously ill practice which they use. They don't sanitize it. They continuously use, and then they touch every non-sterilized items, and then again they prepare it and serve to the people.
Mm-hmm. Good. So, any other participant who want to share their views or their work conducted on such bacteria?
Dr. Prasun sir.
जी sir.
Hello.
जी sir.
So I joined this discussion little bit late.
Yes.
Hello.
Yes sir.
But I'm very happy.
Thank you
I'm in
sir.
Lucknow.
Oh great.
But I've seen many participants have joined. Really, it is a great achievement.
Yes sir.
Many teachers I can see, who are right from Anita, Anupriya, then some PhD people, doctors are also there, correct?
Yes sir.
Dr. Deepika and
Yes.
few more.
Dr.
Prakash
Arul.
Arul is always there.
Yes.
Pardon?
Prakash sir is always there for support. Yes.
Always
Prakash
there.
sir is always there. Yes sir.
Dr. Ashok Kumar Lakshman, you are
Thank you very
which
much.
place? Yes, Ashok Kumar, you are from which place? Dr. Mazid.
Good
Yeah,
evening
yes, sir.
sir.
Good evening. This is Dr. Ashok Kumar Lakshmanan and from Tamil Nadu Vivekananda College of Arts and Science. I
Very
think
nice.
you may know me about myself. Then because then I'm invited you for our then microbiology collegium in this year of the 2023.
Yes,
On
yes.
the time you are then stayed in the Bharidasan University quarters.
Correct. Very
Yeah,
correct.
sir.
Very good.
Yeah, how are you, sir?
Yes, I'm very fine.
Yeah, thank you,
Then,
sir. Thank you.
yes. Dr. Deepika, Dr. Mazid.
Good evening, sir. This is Dr. Majid from Hyderabad, sir.
Yes. You are also regularly attending. Very
Yes, sir.
nice.
Yes, sir. Thank you, sir.
Then Dr. Arzula, something very difficult to
Arul,
pronounce.
Doctor. Yes. So I pronounce him Dr. Arul. He has a
Okay,
long history of
good.
working on such bacteria. Yes, Dr. Arul, are you there?
Yeah. Arul, you are from which place?
Yes, doctor, I am available. I am from Saudi Arabia, King Abdulaziz University.
Yes,
So,
yes.
yeah, I think you know me very well.
Yeah, I know you very well. One
Yeah.
student
Uh,
is one student from India is in need of help. I will write you later on.
sure, doctor.
Okay.
Sure. Okay.
You please say me hello on 98220.
Okay, sure. Sure,
Okay.
doctor. I have your WhatsApp number. I will just put hello message in that. Sure, doctor.
Okay. Okay.
Okay,
Thank you.
doctor.
Thank you. So many
Thank you, doctor.
doctors have joined. I welcome all. And I again request Prasun sir, you can continue the discussion on the Enterobacteriaceae. I will not interrupt more, but I am happy today. That only I will note down. Okay. Thank you.
Yes, thank you so much, sir. Even so, there are several function of these bacteria, and I have so many so many participants have already joined, and some of them are already working, I guess. So, if there are any person who wish to share their views, they are welcome. I welcome Dr. Ms. Anupriya R. Are you there? Anupriya Aqua Bhavani. So, anyways, so as we were discussing that there are some enteric bacteria which are pathogenic. Yes,
I'm Anupriya, sir. You call me.
yes. So tell us something about your work and from which place you are.
Sir, I'm from Karur, sir. Vivekananda College of Arts and Science student, sir.
Okay, great. So you have worked on this bacteria earlier? Yes, thank you, Prakash sir. So some of these bacteria are in the watch list of WHO in the umbrella term that is called escape pathogens. Escape pathogens in which Escherichia coli, Klebsiella, Pseudomonas are the one which belong to this enteric bacteria or Enterobacter family, and most of them are pathogens, and that's why they are in WHO list. Right. I hope some of the participants are already working on it. Anupriya, if you are back, you may tell your views or your work on the same.
Yeah, Anupriya, this is Dr. Ashok Kumar, the head department of biotechnology. I think you know me very well because then previously I'm working as the associate professor in the same UBA department. So you may be the discuss. I think you may the working in an Enterobacteriaceae. It's maybe then you are the final year project, and maybe the discuss your views in this forum, and it may the helping for your then future and also then in your future project also. So you may be the discuss in the about the Enterobacteriaceae.
Yes, thank you, Ashok sir, for enlightening us. And yes, obviously, to all the participants, this is a request that it is a platform of discussion. Maybe your research is going on well, or maybe you may need some kind of inputs from the experts. Like Prakash Alami sir is already there, and there are other uh, doctors, professors, and faculty members who can share their views on your research work. Right. So, Anupriya, uh, Kirtana, Lakshmi, any one of you, if you want to share your views, you are most welcome. Meanwhile, some other fellows start sharing their views. One of the things when I'm repeatedly saying that they are pathogens, this means that they are having some serious consequences after they infect someone's, especially by the action of endotoxins that they release. Endotoxins are the compounds produced intracellularly, but when these bacteria die within our body, it gets released to our bloodstream. And sometimes, if the bacterial load is high, it will lead to a septic shock kind of thing or into toxin shock that may be fatal to the host that is infected with these bacteria. So that's why they are means in the WHO watch list, and they are quite important organism uh, because for one more reason, as they most of them are getting resistant. We are already knowing of antimicrobial resistant strains that are coming up every now and then, from MDR to XDRs. There are a list of strains that are quite strongly resistant to carbapenem antibiotics, our last resort of antibiotics. So this is a kind of last defense that we have against such pathogens, right? And in addition to that, some of these, especially Pseudomonas, they form biofilm. So within a biofilm, they further shows their they can repel or they get a kind of defense mechanism against antibiotic action. Around 1,000 times of the antibiotic dose is required to be effective. So biofilm topic we have covered in some other roundtable discussions, so I will not go in much detail. But to the participants, just to give a background that some of these enteric bacteria are pathogenic in nature and they are being studied so that we can generate novel antibiotics or we can study the mechanism through which they get resistance against these antibiotics. I request Dr. Prakash Alami sir to kindly enlighten us about some endotoxin they produce, this type of bacteria produces, and what are the consequences endotoxin have. Prakash sir, are you there?
Yes, sir. Good evening. This is Dr. Ashok Kumar the Lakshmanan. Before
Yes,
going to
yes,
the
sir.
discuss about the enterotoxins and of the Enterobacteriaceae group of bacteria, is nowadays as a student's point of view, they are the thinking about the Enterobacteriaceae bacteria is only available in E. coli, Salmonella, Shigella, Klebsiella. But apart from that, some of the notable species is also the present in the Enterobacteriaceae group of bacteria in the nowadays is especially in the escape bacteria like the Enterobacter. So the
Yes,
Enterobacter is also is including in the Enterobacteriaceae group of bacteria. And apart from that, some miscellaneous bacteria like the Serratia and the common the gut bacteria is a protease and the Yersinia, the Citrobacter and Morganella is included in the Enterobacteriaceae group of the bacteria. But then nowadays, some of the as a PG level students, they are the thinking about the Enterobacteriaceae group of the bacteria is only like E. coli, Salmonella, Shigella, and the Klebsiella. But most of the students is not aware about the in this type of the notable bacteria like the Enterobacter, Serratia, Protease, Yersinia, and these also then Morganella is also is included in the Enterobacteriaceae bacteria. I think I made the right.
yes, sir, you are very right. And rather, the next point that I was coming about is the non-pathogenic bacteria or some other uses of these bacteria. So very correctly pointed out that as human health is far more important, so people take such work in on priority, and that's why pathogen becomes a research point of interest. From research point of interest, pathogens attract more eyes than some other works. While we know that even in sewage treatment plant, these bacteria are quite helpful, though they are pathogenic if they infect us. But in wastewater treatment plant, such bacteria, especially Pseudomonas or those belonging to Pseudomonas species, they degrade a lot of aromatic compounds as well. I think Dr. Arul has worked on hydrogen production from Enterobacter, if I remember correctly, or maybe Klebsiella strain. Dr. Arul, are you there? Arul sir? So during my PhD, I was actually working on Klebsiella strain as well as Enterobacteriaceae, which was producing quite a good amount of hydrogen using waste like pea shells, potato peels, etc. And we tried to immobilize such culture to increase the hydrogen yield. So they may be pathogenic on one side, but they can also be useful in terms of other aspect such as degradation of polyaromatic hydrocarbons, uh, bioremediation of heavy metals, and biogas production, especially hydrogen. So, any other participant who would like to share their views if they are working on any unique kind of bacteria belonging to this group, they are most welcome.
We can use these bacteria for in microbial fuel cell also to generate bioelectricity
Yes,
from
yes, yes, very correct, very correct. Yes, I missed that part. Very right, Dr.
bioremediation.
Majid.
Yeah. Yes, yes.
So, are you working on microbial fuel cell?
I worked on microbial fuel cell, and it is also published in Elsevier Journal. But
Okay, great.
I used geobacter, not the this enterobacteria.
Mhm.
Mm.
Yes, they have these flagella through which electron transport may happen. It is like it is being reported widely, especially Shigella strain. Some of the Shigella strains are electricogen, if I am correct. They are electrogens. They can release electrons to the electrodes, and that is how the electricity gets produced in the MFC. Am I right, Dr. Majid?
Yes, yes, yes. That's the main point.
Yes, yes. Thank you so much for the input. Dr. Arul sir, are you available? It would be nice if you would share your work on Enterobacter family bacteria. Dr. Arul has a vast experience, especially from bioenergy point of view. He has also worked on MFC.
Yeah, we worked with halophils, especially Acrobactrum, and some of the Pseudomonas strains are there, but still we work with Acrobactrum and some of the Thiobacillus extremophils like acidophilic bacteria. Also, we work with that, and then I think the major part is covered by Acrobactrum in saline under saline condition.
Okay.
Okay, and it is very good in treating the tannery wastewater as well as some of them is efficient in cosmetic as well as this olive oil industries, which has a high salinity up to 6% of saline condition. It was working very well with that wastewater. And also efficiently it was treat up to more than 80% of COD removal and with corresponding energy production was achieved.
So energy in terms of hydrogen, sir, or
No,
the
no, no. It is
electricity?
MFC. You spoke about MFC.
MFC.
Currently,
Yes, yes, yes.
we are working with biohydrogen actually, and we also I think still we are on exploring this bacterial community. One of my PhD students just now she completed. We are in progress of writing the paper. She got some good results.
Yes, yes. I have been following your article, and we were, Miss. I have been following your work, I think, for the past 7 or 8 years, I guess. Yes, in quite similar field. Uh,
Okay.
Great, sir. So mostly you are working on halophiles.
Yeah, halophils. Halophils.
I
Currently, I'm working on biohydrogen production from macroalgae and microalgae also.
okay, because it is the theme there to miss.
Yeah. This is like a next generation fuel. So we have to work on those things and make it like a green energy. So we are in that field. So we have to support.
Okay, great.
Thank you, Doctor.
So, thank you so much, sir, for your valuable input. Any
Thank you.
other participant? Anupriya, you were talking about something, and then I missed you there. Anupriya, are you available? Any other participant who would like to say? IQSC convener has just joined. I would like to know more about their background, and from which institute they belong. IQSC convener, are you available?
Yes, sir.
Yes, please tell us about yourself and what aspect of this Enterobacter suits you in your research or in your study.
Sir, actually, I worked on rhizobacterium, and
Okay,
I am Professor Tabassum from Rampur, UP.
okay. So you worked on Rhizobacterium.
Yeah.
Hmm. So I think so, one of the participant was working on effect of enteric bacteria on pulses, I guess, pulse seed germination. So yes, great. You guys, if you want to discuss, Mariam, I guess Mariam was working on that. Mariam, are you there?
Yeah, I'm here only.
Yes. If you want to share some views, because Rhizobacterium, you may have also worked on Rhizobacterium or
Yeah,
related
actually,
field.
I've worked on rhizobacterium as well. Like my another project was based on uh, isolating pesticide tolerating rhizobacteria from soil.
Okay,
Yes.
okay, great.
So,
You may would like to get in touch with Professor Tabassum. She is already working, and most likely, Tabassum ma'am, can you please elaborate your work? What have you worked on, and what was the output?
Yeah. Actually, we are working for, we are using rhizobacterium as a beneficial thing, you can say, for nematode eradication.
Okay, so it is like mostly for soil health aspect
Yeah,
you have
right,
worked. Oh,
right.
great. They may infect nematodes and reduce their
like
count
my
in the
my
soil.
research was based on like
Yeah.
more on biopesticide production, or
Okay,
because the chemical pesticide harms the crop and therefore affecting the human health.
okay, great. Uh, Renuka Devi has joined recently. Renuka Devi and D. Deepika, are you available? Dhanalakshmi is also there. Participant, we would like to also, those who have joined recently, we are discussing the topic on Enterobacter, and most of them are pathogens. Some of you, Renuka Devi has joined again, I think, from some other device. So this is a
Actually,
platform based. Yes,
Actually, I am more interested to know that what can we do for MSc students as the project for
for
biochemistry.
project there.
Yeah,
Yes.
yeah.
Uh,
Then
one,
please, yeah.
yes, sure. There are so many things actually, and what we were discussing before you joined was that as human health is more important, and most of the funding also goes there. People, it is quite eye-catching as well. So, escape pathogens belong to this group, Enterobacteriaceae, Escherichia coli, Shigella, Salmonella, um, uh, Pseudomonas. They form biofilm. So, One simple project that people can do in any common lab is about antibiofilm activity, as
Okay.
these Pseudomonas and Klebsiella strain they form biofilm, and because of the biofilm they get more resistant to the action of antibiotics. And we all know as a microbiologist that carbapenem class of antibiotics are our last resort. So if we get some resistant or XDR against these drugs, then we are left with no more option of antibiotics. And there are some other treatments that we have to do, which is quite tricky to treat if somebody gets such infection of XDRs. So a simple project can be to look after antibiofilm uh, compounds, so compounds that can degrade or initiate the disruption of biofilm. So if biofilm gets disrupted, now the antibiotics are accessible or they can target the bacteria. So very simple assay that we can do. I hope, ma'am, you know about the antibiofilm activity or the screening assay. Sorry.
Yes, sir.
So in 96 well plate, we can screen several drugs at a time for antibiofilm activity, and the level of antibiofilm can be checked using crystal violet strain. So crystal violet basically stains the EPS, extracellular polymeric matrix, and by the action of such stain, we can quantify the level of biofilm produced. So when you are giving some drug, if the level of biofilm goes down, or the through CBSA, the blue color that will come out, its level goes down. This means less amount of biofilm has produced. So this means the drug was effective. Yes, Bhumika, you want to say something? Bhumika Navina has joined recently, I guess. Okay, so Tabassum ma'am, I was saying that a simple master's project could be. Yes, Dr. Majid, you want to say something?
I would like to add something. Like
Yes, yes,
we can work on this antimicrobial resistant enteric bacteria. We can explore. We can go for the diversity of these enteric bacteria, uh, as well as we were discussing about the hydrogen fuel, right? So
yes,
we can also do the research on these enteric bacteria on bioethanol production, biobutanol production. Even there is more work we have to do with microbial fuel cell even. And in agriculture also we can work like production of biopesticide, or we can check as a biocontrol agent, because
yes,
enteric
very,
bacteria they can also promote the plant health, right? Soil health
yes,
also.
yes, like
And
PGPR activities.
because they produce a phytohormones, so that's help in the plant growth. So we can work on that also. And
That's
there are
great.
so many things to work with the enteric bacteria, right? So
Yes, correct, sir. And among
I
the
think
whatever you,
uh, prebiotics and gut microbiome to treat Klebsiella infections, and then there
yes,
is
yes, yes, that is also there.
issues out there. And there is one more topic which is very interesting, and it was going in my former uni, which was bacteria which can dissolve heavy solids, or that they can tolerate heavy solids found in soil.
heavy solid or heavy
Heavy,
metals?
yeah,
Heavy,
yeah.
sorry, heavy metals. I'm sorry,
Okay, okay,
heavy
great.
metals. Yeah. So they again it is good for agricultural practices.
Yes, yes, very right.
True. Then too, we can do in water also. Heavy metals are present in water. I have seen so many reports which reporting that
Yeah, sure,
heavy
sure.
metals are present nowadays in river water and all. No?
Yes, sure, because TDS is getting higher and higher these days.
Okay.
Yes, and Dr. Majid very correctly mentioned several aspects on which work can be done. And I would like to highlight some of the points. Yes, Mariam, you
Master,
want to?
not doctor. Please.
There is one more technique is there that is used to transfer the enteric bacteria to another person for gut health. That is known as fecal microbiota transplantation. That is also going on. The work is going on that part.
Yes, but I
Fecal
think
matter
it
of
is. Uh,
animals or what?
To humans, to human.
Okay.
yes, but I don't think that
That
involves
is a
enteric
yeah,
bacteria.
FMT. If you can
FMT, yes, yes.
yeah,
I think
FMT.
Prakash Alami sir, if he is here, he will shed
Yes,
more light.
he can put more in.
Mm,
Yeah, maybe in some other programs we can talk on that aspect, because now
yes,
better to discuss with relation to the pathogenicity of Enterobacteriaceae and how it is important from the microbiological or from clinical perspective.
Yes. Yes.
Let us have, and those who are actually working on any of those gram-negative rods, how they are clinically significance with reference to the virulence, with reference to the antibiotic resistance, and their method of detection, perhaps. The classic conventional microbiological method, because this family has been well identified through the conventional techniques, you know, and what are the different tests are available for the Enterobacteriaceae to detect, or even the media. So let us have the discussion on that aspect. We are always there to support and help the people. Now everyone wants to study a gut microbiota or AIML, so that is there, of course. But we should not forget the conventional microbiological approaches also, as well as the tools, because whatever the method is, our gram staining is still actually is required or useful, or physiological microbiological tests are required, especially when we go with the identifying Mycobacterium tuberculosis, right? Yeah, and also let us have continue the discussion, not issue.
yes, sir. Very right. Before I change it to that, sir, for a long time I wanted to discuss about the detection of AMR. So as you all know that this enteric bacteria are mostly gram negative. So can we develop any kind of kit to detect their level, especially the level of antimicrobial resistant bacteria in suppose soil sample or maybe sewage water? So are there any kits available, sir, on AMR detection? They may be detecting enteric bacteria only, maybe the coat protein or the membrane protein of Shigella or Salmonella. I'm not aware. I'm just asking, sir. Prakash, sir?
I think
And
so. Not exactly, because I am also somehow now going more with the molecular tools or bioinformatics tool. But what I understand is that conventional microbiology, with reference to the Enterobacteriaceae, has its own advantages. So,
yes,
just
yes, yes. With
somebody can actually,
conventional by, sorry,
yes,
continue.
somebody can
Mm.
actually put a light on those things. That will be really interesting.
Yes, Dr. Majid, are you there? So there are selective media, as Prakash had rightly said that there are means conventional detection of these microbes are equally important uh, compared to the advanced techniques or advancement in the science that is happening through AI tools or bioinformatic tools, because ultimately we need to characterize these microbes and also uh, to which class they belong so that we can develop respective treatment accordingly. So if it is Shigella, how to approach it. If it is Salmonella, how to approach the treatment. Same holds good for Pseudomonas as well as E. coli. And not all E. coli are pathogenic. So some of the E. coli are means they are just non-pathogenic. They can grow, they can utilize the nutrient, but they are not doing anything to the host, right? And to the previous discussion with Dr. Majid and Tabassum ma'am. So if we are thinking about any master's project, we also have to think about the techniques available. So Prakash sir was actually emphasizing the similar thing that biochemical tests are most common that a PG student should learn so that they can distinguish between two or different microbes. And one thing that I have noticed is that okay, biochemical test, MRBP test, indole test, people do catalyst test also, people do easily, but most of the students they don't learn about the motility assay or biofilm formation assay. Okay, but Kirby-Bor method of antibiotic sensitivity test that is common in the class, but not the biofilm formation. So these things can be incorporated. Means this is mostly for the master's project, not about the research, but short master projects can be developed. And same holds good for the differential media. If somebody can develop a specific media or means selective media for such pathogenic strain or XDR or MDR, that is also a very good theme to work on in a short span of time. Uh, Obviously guided by their mentors.
Yeah, okay, thank you.
Yes, ma'am. And once we are thinking about such master's project, we have to also think about the resources available. So especially for MFC, we need some other kind of reactor. While for gas production or biobutanol, we need to quantify the level of alcohol. For that, we need GC. So gas chromatograph may or may not be available in the vicinity. So such projects may be quite hard for the student to carry out unless we have the facility, right? So while most of the, as I mentioned earlier, most of the thing is uh, focused on health aspect, detection of these organisms, inhibition of the growth of these organisms, or killing of these organisms. So
Right.
yes, so any compound, whether natural or synthetic, that can kill them, selectively kill them. So
Yeah.
that is a point of concern, and that is means a need of the hour basically, because we are losing. I hope every one of you have read the recent report from WHO again. WHO says that we are not having enough antibiotics or antimicrobial compound in our clinical trial. Very recent news it is. We don't have much antibiotics there available for clinical trial. So this means we are not at all having potential leads, and we should always keep on evolving as the bacteria is evolving through resistance. They are evolving themselves. We also have to evolve our antibiotic arsenal so that we can uh, target such bacteria. Otherwise, human health will be in a bit trouble after some time.
Yeah, right.
So
Very true.
then in that case, there is a concept of phage therapy.
Fast
So is
therapy,
it
yes, correct.
yeah? So is it
So
still in trial or is this just a concept right now?
it is a concept so far I have read about it. It is still in the conceptual stage um, because phages have port proteins and phage port protein may be treated for us. So phages are what they identify specific host. Okay, so bacteriophages will only attack bacteria, but once they are in our body or a bloodstream, they will be treated as a foreign particle, right? So they will generate immune response that we may not know. Okay, so
In
people are
laboratory, it is
working.
okay. In laboratory, it is okay. It's working. but we don't know, as you told, that in body, what will be the reaction? Uh, because we have checked this bacteriophage therapy also. So,
Okay, great.
so we exactly don't know how this is in real time, how is it is going to help us. But in laboratory, it is working perfectly.
So on which phase you work, Dr. Majid?
Bacteriophage, actually, we isolated a compound that is that one, not a compound, this bacteriophage named CP113. The paper is already published. You can check
Okay.
that one. Yeah, so it's working well, but we don't know. In laboratory, it's okay, what I can say. But in as a treatment, we don't know how it is going to help us.
Yes, but was this phase specific against E. coli or against Pseudomonas?
It was against Klebsiella.
Klebsiella. Okay, great. So nowadays, combination therapy is also there with phage. With phage, you can add some other thing, and even phages, how they infect the host is through their tail or the so they have depolymerase or enzymes that can degrade the membrane or core protein of bacteria, and that is how phage infect the host, right? By degrading their outer cell and then going to inject the DNA. So this is how they infect. So if we can just use this depolymerase enzyme, we may be able to make a kind of broad spectrum molecule that will degrade the membrane component of the typical pathogen. Okay, this is one of the aspects that people are writing in their article and are also very hopeful that if we can develop such thing, so we may not need phage, we may need this protein, but again, within the body, what kind of reaction is going to happen, we are not aware of it. So if anybody else have worked on such thing, they may enlighten us.
And again, there is also nanotechnology available, but it is not very uh, fast forward in India or any other country. I guess nanotherapy is there, etc. But I guess it is still a concept, and then it is all already situated to lab only.
Yes, with nano thing, na, especially metallic nanoparticles, they are still raising eyebrows in most of the cases because they are made up of metals, and heavy metal toxicity is a concern for us, for animals, because metal can get accumulated and they may hamper our organs. Mm, some of them are fat soluble, so they may be uh, uh, fatty area of our body, okay, including liver or whatever fat do we have in our nearby skin area. So then their accumulation will cause some disease, right? So that's why the metallic nanoparticle people, then people have also developed uh, what chitosan based nanoparticle. There are so many other biopolymers. Their nanoparticle loaded with say antibacterial or antibiofilm compound. People have tested. So these things are in research stage so far I know. Because getting approval from uh, ethical committee as well as the drug has to be approved from the FDA so that you can start working on clinical trials, right? For humans study, human based study. For animal study, it is okay. We can use such nanoparticle, treat the animal, and check for the efficacy of the drug. So we have several participants. Some of them have recently joined. Sharon Oli, are you there? Sharon, Lakshmi,
Sir,
Vinod.
sometimes the pharmacokinetic studies is also the maybe then helpful for the clinical studies. I think so in my knowledge.
Yes, correct, sir. Correct only.
Uh, So I have the one doubt, then because of when the urinary tract infection is caused by the Klebsiella, but we
Yes.
cannot then still cannot then able to treat the Klebsiella pneumonia with the help of the antibiotics, and due to the presence of ESBL and carbamazepines, any the ESBL binders, and it may be then helping for the reducing the then severity of the ESBL, then it may be the helpful for the treating the Klebsiella. If you're having any ideas, it can be then explained in this forum, and it may be the helpful for the future. The because of that's the treatment of the Klebsiella pneumonia, especially in the urinary tract infection patients, that is a need of our now.
Yes, sir. So I don't have much idea about whether someone must be working, but I don't have much idea on it. Maybe some other participants who have been working on this aspect or Klebsiella. Yes, obviously the antibiotic resistance is a very big issue and a serious concern. And even UTI infection is becoming quite general nowadays. And people are coming up, this hospital are isolating every time some kind of MDR or XDR. They are also reporting case studies in clinical journals, both from India as well as abroad.
Yeah, because of the myself, and we are then trying to then finding the ESBL binders, and through the actinobacteria from the past then 4 years, but still we cannot then able to the getting the suitable finders for the treating the ESBL. So that's why then asking this type of the question in this forum.
So, sir, bioinformatic tools, have you tried kind of docking the molecule with the target compound? So there are several. Uh, so recently, I came across a logic that people were discussing in another
Yeah,
forum.
we are also the user of the bioinformatics tools, sir, and then before the 1 year before. But that time is
Okay.
also that we cannot then be able to then finding the suitable finders. The because of the bioinformatics tool, and it may be the helpful for the finding the binders, the component. But then when we are then going to the clinical studies, that
It
binders is
don't work.
the
Yes,
fail.
yes, 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 yes. Got it, got it. So this is how the research goes, I guess. One thing that I was mentioning is recently somebody suggested. Yeah, you may take a note of it if you find this suitable. So what he was suggesting is when we are doing some kind of bioinformatics study, we should use FDA approved drug first. Directly FDA approved drug. We should try to repurpose. So the drug may be effective for some other thing, but since it is FDA approved, so going further with a clinical study will not be much of a problem. And then if you are lucky enough and you get some kind of hit, means the molecule, some kind of molecule that are effective in your case, then you may go ahead with a clinical study. So I means I am not uh, claiming that you will get, but this is one of the approach that people said to do the things fast. You should try for the database that contain FDA approved drugs.
Yes, sir.
And
Thank you. We can try it, and then our the forthcoming trials.
yes, yes, it will basically reduce the time and effort of yours. So you can go to the next step if you are
Yes, sir.
targeting some kind of development of some drug. Yes. Anyone else would like to say something on this? Sharon, Mrinal, Shalini, Sriniti, any of you are available? You may share your views or your work on the platform. Nishika has recently joined. Nishika, are you there? Nishika, madam. Are you there?
Sir, my
So,
name is Mridal. Can you hear me, sir?
yes, Mrinal. Yes, tell me.
Yeah, I've just joined. Uh, Actually, we are into ready-to-use microbiology media plates in the manufacturing of. So we supply these plates to hospitals. Sometimes they find it difficult to isolate Streptococcus pneumoniae and all on blood agar. I
Mm-hmm.
don't know much about its pathogenesis or all. I'm a MSc microbiology, of course, but now I'm into this ready-to-use microbiology plates.
Okay.
So, why is it so difficult to isolate, sir? And sometimes they don't grow. Is it one of the reason that patient must have done self-medication? Nowadays, people do take antibiotics on their own. So, could that be a reason? Sometimes they do not grow on the media plate,
No,
even
growth
if
on media plate can be of various different reasons because growth may not hamper. There are differential media already available, and you may be aware of such composition, maybe from high media. Which company you are in?
I am
Sorry,
into biosmart biologicals.
Biosmart Biological.
Yeah.
Okay. Oh, sorry, sorry. So
Uh,
even high media and this Titan Biotech, there are so many other companies also. They are making selective media ready to use plate basically. And
yes, yes.
even nowadays, kits are also available to distinguish two or more than two different bacteria present in, say, UTI patient sample
Yes,
or
yes.
even that
So
sample.
we do
Yeah.
manufacture different kinds of media, but sometimes Streptococcus pneumoniae is a big problem for them to isolate.
Okay.
What could
Okay.
be the reason?
This is also new to me. Maybe.
Uh-huh.
Uh-huh.
Okay, sir.
But
Okay,
so
okay.
far, so far the growth of the organism is concerned, media are already nutritive, so there may be some other
Yes.
reason of their not
Uh-huh.
growing. Maybe
And
if
it may differ from person to person, that
the
the strain
user
of,
basically
yes,
the user
yeah.
how he is operating, how they are keeping the plates, okay, the moisture
Hmm.
content of the plate. There could be so many reasons. Even the number of bacterial cells present, so the bacterial load. If it is
Uh-huh.
low, then we should keep plate for a bit longer time so that a
Right.
colony can appear.
Hmm.
Hmm.
Right, sir. Right, sir.
But if mostly clinicians need data within 24 hours.
Yeah, madam, it's a
Yes,
Streptococcus
24 hours.
pneumoniae. It's too difficult to culturing, and then because of it's due to the pneumolysins. So the because of its
Uh-huh.
presence of the pneumolysins, and it may be is chances to the quorum sensing. So in this reasons, and it's difficult to the culturing on your normal agar plate.
Okay. We are growing it, we supply blood agar plates for that, especially, and Miller Hinton with sheep blood. So sometimes they find it difficult. So maybe that enzyme may be the reason, the pneumolysate. Hmm. Okay, so that was just my query. I can't give much inputs on the other topic. I just
No,
joined.
this is rather this is the platform where we can discuss such thing. And if
Yes.
suppose there are such issues to the industry or the user, then there is a scope of
Hmm.
research as well, right? So maybe some of the participants may already be noting these things.
Yes, yes, sir.
Yeah, Dr. Prasann, actually what madam is talking, is a very valid point, and this is where the actually industry will be facing those challenges, which
Yes,
we
sir.
Yes,
will
sir.
not be aware, or you will not get those information anywhere, you know, because
Hmm.
Yes.
Hmm.
some of this kind of data will not be published also, you know.
Hmm.
So they will just keep with them in a confidential way. And then usually becomes actually a challenging, especially you look for a culturable methods or looking for cultivating those bacteria.
Yes.
And then not every time that your molecular methods will work. Molecular
Right.
methods will only tell yes or no, but what is the next? So if you want to do the additional studies, then you need the microorganism has to be cultivated properly, has to be identified. It has to be grown and maintained also. And
Yes,
then this
and then
actually
they have to do, yes, the
practical
antibiotic
experience.
resistance.
So yes, exactly. So if you are doing suppose antibiotic resistance or virulence test, then you have to cultivate that organism. And
Right.
then only we will be able to further take up the additional studies, you know. So I think that way our microbiology society forums actually is doing very noble cause and bringing those
Yes,
expert
sir.
from within the microbiology, those who works in the different areas. We should actually look more from the industry perspective. I think subsequent
Yes,
discussion.
sir. Yes.
Yes,
Yes,
and
there are
rather we
many
Time
should
in
ask.
fact,
challenges that
I
we
told
face.
Deshmukh sir that we should have more lectures on the industry and microbiology. Like
Right.
Yes,
mushroom.
exactly. I wanted to tell that yes, Mrinal madam should join our forum
Yes,
quite
yes,
often.
yes.
Yes,
And she should give the emphasize on what are the difficulty or challenges they are facing or when she is observing, so that it is useful for us also to give a thought. Because very initially I actually pointed out that not every time molecular techniques or NGS will be useful. And then you see that this is actually how the cultural methods are important. Is a very live example, you know that if you have to go for the serology or if you have to go for the virulence factor determination or the antibiotic sensitivity assay, so you have to cultivate the organism. Then only you will be able to answer. You know, so we have to actually parallelly need to rely on the our conventional microbiological tools also. And what you actually pointed out related to the flagella is very important because that is associated with the mobility, right? So we should know how the flagella staining can be done and then motility can be determined. Very important aspect on that also.
yes, Pavitra. Pavitra, you want to say something? So very rightly said, sir. Pavitra, can you mute yourself?
Somebody wants to share their personal discussion also, I think, on this group.
Yes, I'm muting them, but it is getting unmute. Sorry.
You can have that mute all function. Maybe that can actually answer. And then you
Yes,
can
sir.
give the, okay, now it looks quite all right. Okay. Somewhere little bit entertainment also is required.
Correct, sir. See, I
Mm.
have muted guys, but still it is coming up. Pavitra, are you listening to us?
Sir,
Pavitra, you want to say something?
no, sir.
Then whose microphone is there? Tamil. Tamil Selvi, please unmute yourself, please. Yes, thank you, Prakash sir, for this insight. And yes, I think industry fellows, this is a very good platform. And if I summarize today discussion, we got two or three unique topics or unique thing out of it. One is from recently from Renal Madam about the detection issues, especially the growth issues of uh, Streptococcus, right, ma'am? Renal Madam?
Yes, sir. Streptococcus pneumoniae.
Pneumonia, right? Yes. And then you, sir, you also, Ashok, sir, you mentioned about uh, how to generate the lead molecule, how to detect, means screen for the lead molecule against the virulence factor or about the resistance, how they get resistance. So can we develop a lead molecule? And then the master work I think Professor, sorry, I forgot the name, ma'am. So master's thesis topic because these are the manpower, future manpower who will get trained on such theme. So this was quite a fruitful discussion, and two or three things we can carry forward. Yes, Mrinal ma'am, you wanted to say something.
No, sir, just I had unmuted, but you got the name, sir. Streptococcus pneumonia I was discussing about that. And I'm very glad that I have got this platform to discuss the issues in industrial microbiology, especially clinical microbiology. I'm
Yes, yes,
really
yes.
hopeful
And
that it's going
this,
to be of great help.
yes, yes, this is also a food for thought for us as well, right? Because
Yes,
we
and
may
it
be sometimes
enhances
just
the
focused on,
knowledge, sir. Otherwise, we are into industry and it becomes monotonous. We
correct,
don't
correct.
know what other things are going on.
Yes,
So it's
yes.
a
And
very good platform, sir. Thank you so much.
yes, ma'am, and we would like to request that please join the session because at least from industry aspect, things you may discuss whatever is there to in your knowledge, at least, right? So this will help
Yes,
all other participants.
yes, yes.
Yes.
We
So
can
with
discuss
this,
about the
We,
different
we can
media
conclude now.
and all. Yeah.
yes, ma'am. So with this, I would like to conclude today's session, and I thank all the participants, Dr. Prakash sir, Renal madam, uh,
Thank you.
and
Thank you very much.
yes, yes, Dr. Ashok sir, Dr. Arul sir, and all other participants, Mariam, uh, and one ma'am from Lucknow. I forgot the name. Sorry, ma'am. So, but you all
Professor
have participated.
Tabassum.
Yes, Tabassum ma'am. Sorry.
Thanks.
Yeah.
Yeah, thank you so much.
Yes, yes.
Yeah,
So
it is
thank
really
you all
fruitful.
for joining in, and we hope your active participation in the subsequent session as well. It will help us grow as well as learn together. While
Definitely.
the student participants will learn more on the recent topics that are not
Exactly.
yet
Yes.
touched or that needs more attention of research, right? So with this, I would like to thank you all. Thank you,
Wonderful.
Prakash sir.
Thank you, everyone.
Yeah.
Sir, can we get the certificate of participation or
Great, great, great.
anything?
Thanks for reminding me. Thank you so much for reminding me. I am sending you the link. You all
Mm-hmm.
participants are requested to fill this feedback form. Thank you so much
Yes.
for reminding.
Yes.
Yeah,
Yeah.
Thanks.
yes, yes.
Okay, sir.
So I
Yeah.
have sent in the chat box. All participants are requested to fill this form to get the certificate. Those who will fill this form, they will only be able be recorded, so they will get the certificate. Okay, all the participants
Yes.
are requested to positively fill this form before
Thank you,
moving on.
everyone.
Yes, thank you. Thank you so much, sir. Thank you for the help.
Thank you, sir. Thank you. Thank you.
Okay.
Thank you,
Thank
sir.
you. It
Thank
was really fruitful.
Yes, welcome. Renal ma'am, are you there? Mrinal madam.